Postpartum depression, often referred to as PPD, is a serious mental health condition that affects many women after childbirth. It goes beyond the typical baby blues, involving persistent sadness, anxiety, and fatigue that can significantly interfere with daily life. These symptoms can manifest in various ways, impacting a mother's ability to bond with her baby, care for herself, and manage daily tasks. While the exact causes of PPD are complex and multifaceted, recent research has shed light on the neurological changes associated with this condition, particularly in relation to brain regions like the amygdala and hippocampus. Recent advancements in neuroimaging techniques, particularly magnetic resonance imaging, or MRI, have enabled researchers to delve deeper into the brain's structure and function. A groundbreaking study published in the journal utilized MRI scans to investigate the brain changes associated with PPD in first-time mothers. The study involved scanning the brains of 88 women during late pregnancy and again shortly after childbirth. A control group of 30 women who had never given birth was also included. This allowed researchers to compare brain changes in mothers to those who had not undergone pregnancy and childbirth. The study revealed intriguing insights into the neurological adaptations that occur during pregnancy and the postpartum period. Researchers observed that certain brain regions, including the amygdala and hippocampus, underwent shrinkage during pregnancy. This shrinkage was particularly pronounced in areas involved in social cognition, suggesting a potential fine-tuning process that prepares the brain for motherhood. This fine-tuning is believed to facilitate a mother's ability to interpret her infant's cues, foster a strong maternal bond, and navigate the demands of caring for a newborn. The amygdala, a small almond-shaped structure deep within the brain, plays a crucial role in processing emotions, particularly fear and anxiety. In the context of PPD, researchers discovered a compelling link between the volume of the amygdala and the severity of depressive symptoms. Women who exhibited more pronounced increases in amygdala volume during pregnancy were found to experience more severe perinatal depression symptoms, both during pregnancy and after childbirth. These findings suggest that alterations in amygdala volume could serve as a potential biomarker for identifying women at a higher risk of developing PPD. This discovery holds immense promise for developing more effective diagnostic and treatment strategies for PPD. By identifying women at risk early on, healthcare providers can implement preventive measures such as closer monitoring, early intervention programs, and targeted therapies to mitigate the severity of depressive symptoms. Section 4. The hippocampus linking birth experiences to brain changes. Another key area of interest in postpartum depression research is the hippocampus, a seahorse-shaped structure involved in memory and emotional regulation. Interestingly, the study revealed a correlation between hippocampal volume changes and the nature of a woman's childbirth experience. Women who reported more difficult or stressful births exhibited greater increases in hippocampal volume after childbirth. This suggests that the brain may undergo structural adaptations in response to the challenges and emotional intensity of labor and delivery. While the precise mechanisms underlying this correlation are still being investigated, one theory posits that heightened stress during childbirth could trigger the release of hormones that influence brain plasticity, leading to structural changes in the hippocampus. These changes may reflect the brain's attempt to adapt to and process the emotional impact of the birthing experience. Section 5. Implications for diagnosis and intervention. The findings from this study have profound implications for our understanding of postpartum depression and its underlying neurological mechanisms. Firstly, the identification of brain regions associated with postpartum depression opens up new avenues for diagnosis and early intervention. By monitoring changes in amygdala and hippocampal volume, healthcare providers may be able to identify women at a higher risk of developing postpartum depression and provide timely support. Secondly, understanding the role of birth experiences in shaping brain changes emphasizes the importance of providing comprehensive support during labor and delivery. By minimizing stress and promoting positive birthing experiences, healthcare providers can potentially mitigate the risk of postpartum depression and foster healthy maternal mental health. Finally, this research underscores the need for continued investigation into the complex interplay between hormonal fluctuations, brain plasticity, and environmental factors in the development of postpartum depression. As we gain a deeper understanding of these intricacies, we can develop more targeted and effective interventions to support mothers and ensure their well-being during the postpartum period.